We've just finished up the first of the Soapbox Science and Arts event here down in Oxford, and you've literally just stepped off your soapbox. So how was it?
It was exhilarating. It was really challenging, but also really cool. I love the interaction and the engagement we get from the audience at these events.
So tell us a little bit about the sort of research that you were talking about.
Okay, so I do atmospheric dispersion, and in specific I'm looking at biomass burning in Southeast Asia and how that impacts the air quality in Singapore. And I was using soap bubbles to illustrate how particles disperse in the atmosphere, which is really cool to get all the kids involved.
Absolutely. Bubbles will always draw the kids in, although in some cases the adults as well.
Yeah, which is great, which is awesome, right? The kids get to come out in all of us. So I was also using, from my artist, she constructed experiments sort of with vases, and she'd built these Lego villages that we put in the bottom of the vase and then had a lot of water, and then we were dripping food dye in these while people were using straws to actually generate wind and movement in these vases.
Oh wow, okay.
And that way we were able to illustrate how movement in the atmosphere and the dispersion of this food dye.
So audience participation to demonstrate what was going on in respect to your science.
We could, yeah.
Okay, excellent. And how did it go down?
It went down really well, I think. We got it in lots of kids and also some grown-ups, even to glow soap bubbles.
Oh, nice. That's great to hear that they were getting involved. Yeah. So whose idea was this? Was this something that you came up through chatting? Were there a lot of dead ends? Or was this an obvious one to be able to illustrate how your research works?
So the soap bubbles are actually my idea from last year, so a bit of recycling. And the vases were all Kate's idea. She found some diffusion experiments on YouTube, and we adjusted them to be dispersion rather than diffusion.
Oh, that sounds like a great collaboration between the two of you. So
really
you've done a regular soapbox, and now you've done a soapbox science and arts one. Which one do you prefer?
In some ways they're different, but in some ways they're also quite the same. I think in my previous soapbox I put a lot of emphasis still on audience participation and interaction, and also having other experiments. It's definitely, for me, it feels more like you have more responsibility, right? Because you want to perform well for yourself, but you also want to perform well for your artist.
Absolutely. But so with respect to your research and getting your message across, which one do you feel did it better?
I think they both did really well, actually.
They both did it really well?
Yeah.
Okay, it's a toss-up on this one, guys. It's a toss-up.
Our atmosphere will be outside.
I am Cristiana Magnoni. I'm a second-year PhD student here in Oxford University. I'm based in DPAG, that is the Department of Physiology, Anatomy and Genetics, and I study the connectivity of brain development, so how brains form its connections during the first weeks after birth. Today I try to explain my technique using an analogy with battleship. So essentially, what I'm telling people is that I play all the time with neurons to find hit and misses and actually understand how neurons are connected, as if I'm playing battleship, but instead of looking for battleships, I'm looking for connected neurons. And so a hit is a connected neuron, a miss is a not connected neuron. I love public engagement. I am doing a lot, and I found it's really nice to go out of the lab and explain what's happening in the labs, because essentially science is not for scientists only. So there is so much science around our everyday life. And a lot of this science is actually funded by public money and by charities. So I think that the public itself has the right to know what's happening, and we as scientists have the responsibility to tell them what's happening. But actually this kind of event in which you merge science and art, with this event you are able to reach all these people that maybe are just going for the art bit and say, oh, actually today I also learned something else. And they will be less scared to approach science.
An awful lot of species that are all dependent on each other, and it's very, very fragile. So within those species you'll have, they'll all have a role that keeps that ecosystem healthy. One good example is parrotfish. They're grazers, so they eat the algae from the tops of the corals. And in doing this, they keep the coral algae free so that sunlight can get through and coral can continue to grow. So what I do is I look at the problem of fisheries in a socio-ecological context, and that's studying the whole system of people and wildlife together. And in that process we have to start concerning ourselves a bit more with human behaviour, the drivers of poverty and the drivers of why someone would choose to fish badly, knowing that it might mean that their children won't have the same resources in the future.
I'm Lucy Mapp. I'm based at the University of Southampton and I'm doing my PhD in co-crystals and crystallography. I'm looking at how we can change properties of drug molecules by adding in other molecules into the crystal structure and how that relates to their properties.
I'm Tess Tallulah and I'm studying fine art at Oxford Brookes down the road. I was really interested when I heard that you're studying drugs and crystals. That was all I got at first, so I was really intrigued.
Drugs are generally made of crystals, chocolate's a crystalline material, and I look at how the molecules in the crystals pack together and how that affects their properties. And so you can relate different forms of chocolate. The way the molecules crystallise in the chocolate gives you different forms and different textures, and then that's all related to structured properties. And it's exactly the same principles with drugs, how the actual molecular structure and the way the molecules pack together affects their properties. And I tried to modify these properties by adding in other molecules to change them, to make them either more soluble, less soluble, have faster dissolution, change their melting point, depending what's required for the ideal drug for that situation. And if you start telling people there's different forms of chocolate, they go, ooh. And I'm not talking about white, dark milk, Cadbury's Galaxy, Nestle. There's 6 different ways the molecules can crystallise together, and there's only one which is actually made into the manufactured version of chocolate, which has that nice smooth texture, melts in your mouth, and all the properties which gives you that nice chocolate bar at the end. Different drugs will have different requirements, so you might want one which you take it and it's got a very fast action or fast release to get to the site of action much more quickly, so you've got a headache, you obviously want that to go away as quickly as possible. And there's other diseases where you might target it with a drug, but you want it to slowly release the drug molecule over a sustained period of time. I think it's a really great combination of having me as a scientist trying to talk to the public, but having some sort of in between in terms of the art form, where I've explained what I do to an artist who's taken a completely different way of thinking about it, because scientists often have very hard, half-assed ways. This is right, this is the science, this is this.
Yeah.
And you just mention a few things, and artists are generally much more kind of free-flowing and have different ideas and concepts. Just to get different people's perspectives and different ways of conveying certain things. I've sort of heard quite a lot of different things from scientists. They've interacted with different areas. Someone did it kind of through music and sculptures, and I think that's really great because scientists have their set ways that they produce things. It's generally journal articles and written, and that's not a good way to get anyone who's a non-scientist or even a non-scientist in that area to learn about what you're doing. And if you can engage in any other way and get it across, it'll be great.
I was talking about how we can save the oceans from a thing called ocean acidification, which is where the ocean's pH is dropping because of the increase in CO2 in the atmosphere. A specific way that I look at is by adding this crushed up olivine, silicate-rich rocks to beaches, and that basically enhances a natural weathering reaction that takes up CO2. What I've got here is called universal indicator solution, and it changes colour depending on how acidic the solution is. So if it's really acidic, it will go red. If it's really alkaline, it will go purple. If it's neutral, it'll, like water is, it'll go green. So what I'm going to do, I'm going to keep this one here for reference. So I said CO2 makes the oceans more acidic. We breathe out CO2, so the easiest way to add CO2 to the water is to blow bubbles into it. Do you want to do that? Do you want to be my volunteer? So it's going to make the water more acidic, which will make the solution go from green to yellow to kind of orange to red. But you can kind of see it's going yellow already. And if you keep blowing,
totally. We've had a clear colour change there if we compare that one.
So
That's quite impressive.
yeah, you can just see how quickly the CO2 has changed the pH. This sediment, which is almost entirely olivine, crushed up, and then if we add it to this, even though it's cloudy, it is green. It's gone instantly.
It returns back to green. How did your artist tie into this then, to demonstrate this?
So when we first emailing, she really liked that idea of colour change. I gave her loads of pHs around the world. So we've got a picture representing the Pacific Ocean in 2005, and then this is the same ocean, same spots where the data was collected, but it's 2015.
But we can see that we've got quite a clear kind of colour change in these.
So this is an exaggerated scale, but we wanted to exaggerate it to show that a change of 0.02 is actually quite significant with regards to oceans and these creatures. They don't like a change of 0.02.
So it's a very visual representation.
Yeah, I think it works.
Yeah?
It ties in really nicely with the little experiment I did.
So an enjoyable experience?
Definitely an enjoyable experience. So like I said, I'm from Cardiff anyway, and I'm definitely going to see if they want to bring that art. They've got Soapbox in Cardiff, but they haven't got the artist involvement, so I'm going to try and push for that, because I think it was I really enjoyed working with my artists. So.
Fantastic.
Yeah.